My name is Marianne Chapman and I'm an intensive care clinician at the Royal Adelaide Hospital, and I also have an affiliate clinical title with the University of Adelaide. So I'm one of a team of researchers in the ICU in Callan, and my main area of research interest is gut pathophysiology and nutrition in the critically ill. And hence, I'm also part of a group of researchers that form the Centre of Research Excellence in Translating Nutritional Science into Good Health, which straddles across the University of Adelaide, the Royal Adelaide Hospital and SAMRI. So the Nutrition CRE is made up of a number of highly successful and well-known investigators, and I'm pleased to be able to introduce two of them to you today. So our first speaker will be Professor Chris Rayner, who is a clinical and academic gastroenterologist in the Royal Adelaide Hospital and at the University of Adelaide. He was one of the founding members of the CRE, and he's going to give us an overall view of the work that's currently being undertaken by the group. Thanks, Chris.
Well, thanks very much, Marianne. I'll just share my screen here. So it's a pleasure to be able to talk about our research centre today. The history of this centre really is that in the 1980s and 90s, Adelaide became known as a global leader in gastrointestinal function studies, and that was not in least part due to the efforts of these two individuals. John Dent, one of our former directors of gastroenterology, pioneered the use of silicone catheters containing multiple lumens to measure pressures at different points in the gut and infuse nutrients or other substances to specific regions. And Michael Horowitz, still known to many of you as an endocrinologist, used nuclear medicine techniques to look at gastric emptying and gut transit. And so it's no surprise that a gut focus is very much at the heart of our philosophy about nutrition. And it makes a lot of sense. If you think about eating a meal, you've got gastric distension going on, stimulating vagal afferents. You've got emptying from the stomach at a tightly regulated fashion into the small intestine. And it's here that a complex array of nutrient gut interactions take place that involve hormone secretion and also neural pathways. And these in turn create feedback on gastric emptying and gut motility, on insulin secretion, and on appetite control. And so this is very much the philosophy that our centre was set up with. We were funded under the NHMRC Centre of Research Excellence scheme initially in 2007 for a 5-year term, and we're one of only, I think, 3 centres nationally to have had a second 5-year term of funding starting in 2012. And I think the aims of this scheme really exemplify a lot of the goals that we're being asked to reconsider with all the research reviews that are going on at the moment. So we aim to generate new knowledge, but also to translate these outcomes to policy and practice. We also aim to develop the medical research workforce and to foster collaboration. And if you look at the list of the investigators currently associated with our centre, I think it will strike you what a cross-disciplinary group of people we are. And this is an ever-changing mix of people. People have come and gone over the years, but you can see we've got medical specialists in gastroenterology, a big focus on endocrine, but also intensive care. We've got allied health input, dietetics, we've got medical imaging. We've also got a recent addition of oral health. Together with that, we have nutritional scientists and basic scientists. And so you can see that we're enmeshed in the clinical activities of the Royal Adelaide Hospital, but also in the basic science capabilities of SAMRI and in the clinical capabilities of the AHMS building of the University of Adelaide. And so using concepts that we've developed both in the basic science lab and in the clinical laboratory, we're then able to pursue a range of studies which enable us to get outcomes in the area of optimizing blood glucose in diabetes, weight loss and maintenance in obesity, optimizing ICU outcomes through provision of good nutrition, and preventing morbidity and hospitalization in the aged population. The sort of tools that our centre has to offer include those that look at transit of gut content. So I've spoken briefly about manometry and scintigraphy, but we also use a lot of ultrasound. We use stable isotope breath tests. We use a technique of impedance to look at flow of content in the gut. But we can also deliver nutrients to specific sites in the gut to understand the physiology. We've got a gut peptide laboratory to enable us to do these assays at an economic cost. We've increasingly turned to some endoscopic techniques to biopsy the mucosa and study expression of receptors and transporters related to nutrients. And we can measure gut sensations and energy intake. So how do we collaborate and foster this kind of research pattern? Early career researcher mentoring has been an important focus and has been a formal goal of the centre. One of the really important things that we brought in right at the start and have kept going is what we call the Wednesday Wrap, where we get people either within the group or visitors from outside who come along and present concepts, often unpublished data, maybe a grant application that's in preparation, and we get widespread cross-disciplinary feedback. As I've talked about the gut hormone laboratory, where we have 2 technicians that work on this analysis. And it's been a fantastic focus to bring renowned international visitors who often stay for a week or 2 and have conversations with a wide variety of people in the group. Of course, to run this kind of thing, you need someone who glues everything together. And for us, that's been our project officer. And currently we're thrilled to have Fidelma McGorry as our project officer, and she's an essential member of keeping everyone together and keeping all the nuts and bolts running. And one of the really other precious assets of our group has been our biostatistician, Kylie Lang, who's been with us almost from the start. And to have a statistician in the group that really understands the kind of work that we do and we can go to at the drop of a hat to help solve problems, but also, of course, plan studies, has been a huge asset for the group members. So I'm going to show you just a couple of snapshots of the kind of things that we do. And here's some work that I've done in collaboration with Richard Young, who's a basic scientist working in SAMRI. And this is really on the theme that the gut throughout its length contains receptors for nutrients, which are the same as the taste receptors on the tongue. We've focused particularly on sweet taste receptors, and we now know that these are a T1R2, T1R3 heterodimer. And they exist on scattered cells in the intestinal mucosa. Here you can see an immunostain of a duodenal biopsy that we took from a human subject. You can see where it's stained for T1R2 and that there's a relationship between that and the expression of SGLT1, which is the major glucose transporter in humans. And we feel that to the sweet taste receptor, one of its functions is probably to upregulate glucose transport in settings where there's a lot of sweet substances around. So this quite likely links into explaining why low-calorie sweeteners don't seem to diminish the risk of diabetes with sweetened beverage consumption. In fact, it's just as bad as sugar-sweetened beverages. And with Richard, I undertook this study funded by NHMRC, looking at the effects of low-calorie sweetener supplementation. And after only 2 weeks, we found a substantial increase in glucose absorption in the gut. We found increases in the plasma glucose response to a standardized glucose load. And we took fecal samples from these volunteers and looked at the microbiome. And after only 2 weeks, there were shifts in the kind of organisms that were present. So a reduction in the commensals and an increase in potential pathogens, which potentially drove some of these metabolic effects. Here's another snapshot of Christina Finely-Bissett's work. Christine has been looking at using very low doses of macronutrients to target some of the physiological mechanisms that I showed on one of my early slides. So here she's used a C12 fatty acid lauric acid or an amino acid tryptophan and given very low doses through an intraduodenal catheter. And you can see in these healthy subjects that the fatty acid and the amino acid have stimulated the satiety hormones CCK and GLP-1. they've suppressed the hunger hormone ghrelin, and the combination of those two macronutrients, even in a very low dose, was enough to reduce energy intake in healthy volunteers by about 20%. So a lot of potential in that work. Here's another snapshot of some work where a basic scientist, in this case Frank Grutzner, who's from the genetics department at Adelaide University, and he's an expert on monotremes. Now, I talked about the hormone GLP-1, glucagon-like peptide 1, and some of you may know that a very similar peptide was isolated from a lizard, the Gila monster. And that compound, exendin, was the basis for exenatide, which is now commercially available treatment for type 2 diabetes. Now, it turns out that Frank, working only a short distance from us, was isolating a very similar GLP-1-like peptide from monotremes, the echidna and the platypus. And with MedVet funding, Frank's now working with members of our group to look at developing this compound further and look at whether it could be a commercially viable alternative treatment for diabetes and/or obesity. So I'm going to wrap up there, but I think it's important to acknowledge some of the huge advantages that we've got working in the biomedical precinct. In the University of Adelaide, the sort of physiological studies that we do would be impossible without our clinical research facility. It contains, amongst other things, a dedicated gamma camera that we can use that's not dependent on finding time in amongst clinical studies. The Royal Adelaide Hospital has been a fantastic place to collaborate into. I particularly want to mention the investigational drugs pharmacy headed by Peter Slobodian, which has been a terrific partner in getting a lot of our studies up and running. We're very grateful that even though there's a general lack of research space in the RAH, the Park Clinical Research Facility has been a venue where we've been able to do some more invasive studies, and that really is an essential part of a teaching hospital. The endoscopy unit's been terrific about collaborating with endoscopic studies. And I think it's fair to say in Adelaide that our Human Research Ethics Committee has been gifted by having a series of chairs who've been highly pragmatic, and the regulatory environment in Australia is a real boon for doing things that can't be done elsewhere in the world. From a funding point of view, we're indebted to the Hospital Research Foundation, the RAH Research Fund, Diabetes Australia and Diabetes SA, and of course for major funding, not just for the CRE, but for the projects that we undertake within it, the NHMRC has been a huge contributor. So we certainly welcome as a centre contact with anyone who's interested in finding out more or indeed collaborating with our centre, and there's the contact details for our project officer, Fidelma. So thanks very much, Marianne, and I'll hand back to you at that point.
Thanks, Chris. So I'm just looking at the Q&A section, and there are not any questions right at the minute. So we'll just give people an opportunity to type in anything now. And I'd just like to ask you, so you've got a very diverse range of example projects that you've described for us there, and a lot of them have implications for the general public or for a large group of patients, for example, the diabetics. So in that sort of research, how do you translate that out into, for example, dietary change within the, for the general public? Have you got ways of communicating with them? Yeah, if you can tell us how the CRE goes about doing that.
Yeah, so that's an excellent point, Marianne. To use an example of that low-calorie sweetener work that we're doing, one of the things that Richard has done, and I've done more recently on a grant application, is to look at who we've got that can translate some of those concepts into public health policy. And we have linked up with Caroline Macmillan in the public health area, who's got expertise on policy in regard to tobacco legislation and more recently sugar sweetened beverages. And that's the kind of linkage that if we can find positive outcomes from our currently funded work, we'll be able to take those findings and leverage them into public health policy. I mean, there's so much work around sugar taxes and that kind of thing at the moment. If we can establish that potentially artificial sweeteners are as bad as sugar, we need to be adjusting policy to look at alternatives.
Yeah, absolutely. Now, there is a question that's come through. I think it's a question. It finishes with a question mark. So no collaboration with FMT microbiome people at the QEH question mark. So that's Costello et al. So I guess they're asking if you have got any collaborations with that group.
Look, I've got as a gastroenterologist, have a lot to do with Sam Costello and Rob Bryant and the team out there, and we've certainly talked about a number of projects from the microbiome point of view. Gerrit Rogers, who's in SAMRI, has been a key collaborator to do some of those microbiome studies that you've seen. But absolutely, you know, if we wanted to look at some point about doing some FMT work based on some of the beneficial organisms that come out of that work, Sam Costello would be the natural collaborator for that kind of thing.
Okay, all right. All right, well, thank you very much, Chris. So I think now we'll move on to our second speaker. So our second speaker is Dr. Lee-anne Chapple. So Lee-anne is a clinical and academic dietitian. She's based at the Royal Adelaide Hospital and at the University of Adelaide. She has a current NHMRC fellowship, and she leads the nutrition-related research which is currently being undertaken in the ICU at the Royal Adelaide Hospital. And Lee-anne is going to tell us a bit about that. Thanks, Lee-anne.
Thank you very much, Marianne. Just share my screen. All right, hopefully everyone can see that. So thank you very much for the invitation today and for everyone for joining us. My presentation will be delivered on behalf of the ICU research group at the Royal Adelaide Hospital. So who are we? So our research group is embedded with the Royal Adelaide Hospital Intensive Care Unit. This is a 48-bed unit that has over 4,000 admissions per year and is the major trauma referral centre for the state, being one of the largest and busiest ICUs in the country. Our group is led by Professor Marianne Chapman and Professor Sandra Peake at the QEH and is a diverse group of clinical academics, including nurses, intensivists, dietitians, and higher degree research students. We collaborate widely within the Royal Adelaide Hospital, including disciplines and departments such as gastroenterology and dietetics. And nationally, we're affiliated with a range of different hospitals and institutions, such as the Australian and New Zealand Intensive Care Society Clinical Trials Group. Within the BioCity precinct, we are affiliated with all three of the universities as well as SAMRI, and we have a range of different international collaborators, just a few of which are shown here. These people have been selected for their diverse expertise, such as body composition, muscle metabolism from Maastricht, and respiratory physician from the UK. We also have a number of industry collaborators, such as those that develop nutrition products, pharmaceutical products, and equipment, including Cardinal Health, for whom we're trialling post-pyloric tube insertions at the moment. And we're very thankful for all the financial support we've received from a number of different institutions. So what do we research? Basically anything ICU. This is just a list and not a complete list, but we study anything from neurotrauma to sepsis to the microbiome. Our main focus, or one of our focuses, and what the theme of the presentation today is nutrition. So what nutrition research do we do? Well, by way of background, we know that around 50% of patients are malnourished on admission to intensive care, and they lose around 30% of muscle mass in just 10 days in the unit. This has been associated with poor functional recovery, with patients showing poor function that persists even 5 years after they leave ICU. However, a number of barriers exist to feeding these patients appropriately, such as delayed gastric emptying, malabsorption of nutrients, fasting for medical procedures, being the sickest patient cohort in the hospital, and they have elevated nutrition requirements, which make achieving difficult. So I'm just going to go through some of the research that our group has been doing to help. So one of the specialties of our unit is complex physiological trials. This example was a crossover trial in 20 critically ill patients that looked at the effect of enteral nutrition formula, being an energy dense or a standard enteral nutrition, on gastric emptying. The study design for this came from a clinical question from practice. This was made possible with our links with the CRE, in particular Professor Karen Jones, who was a nuclear medicine technologist, and funding support from the University of Adelaide Ian Potter Foundation and the Hospital Research Foundation that enabled us to purchase this gastric emptying technique, which was a portable gamma camera. So the portable gamma camera allows us to measure gastric emptying at the bedside in these critically ill patients, and we're one of the only units in the world that has this capability. We've also recently conducted a complex physiological study looking at protein metabolism in critically ill patients compared to health. This was done in collaboration with Professor Luc van Loon and Dr. Imre Kauw from the Netherlands. And Dr. Kauw is now completing a postdoctoral fellowship within our group funded by a Dutch fellowship. This was also made possible with support from the NHMRC as well as the Royal Adelaide Hospital Clinical Trials Pharmacy. With Professor Van Loo's help, we were able to purchase this intrinsically labelled protein that was developed in the Netherlands, and we fed that duodenally to our volunteers. We could then measure where this protein goes, both from plasma samples and from a muscle biopsy that was taken at the bedside by our ICU registrars. This study showed that the digestion and absorption of protein was similar between the two groups, but ICU patients had a 40% less incorporation of protein-derived amino acids into skeletal muscle protein. These findings are really novel and show that we need to come up with different strategies to help our patients absorb and utilise this protein correctly. This study forms just one of many that we're doing into research around protein to improve clinical outcomes for our patients, and I'll go through a few more now. So some of our work has been looking at multi-centre point prevalence studies that are used to quantify current practice to assist with trial planning and to see how effective our trials are in being incorporated into clinical practice. These have been taking place in 2012, 2018, and we're repeating again this year in around 40 Australian and New Zealand sites and over 500 critically ill patients at each time point. Between the last two surveys, we showed that there was a greater prescription of higher protein formula in these ICUs in Australia. These results have been teased out a little further by one of our PhD candidates, Dr. Tej Murthy. She collected data at a number of sites in 120 critically ill patients and showed that while the mean protein prescription to these patients was within international guidelines, the amount that actually received was much less at 0.8 grams per kilogram per day. We've also conducted a systematic review and meta-analysis. This one was led by Mike Davies, who is now a consultant within our group, and looked at studies that provided 2 different protein doses. What this showed was that protein dose did not affect mortality, but the dose even in the higher group was well below international recommendation. So there's still a question to be answered here. This has led us to conduct a double-blind randomised feasibility trial. This one, called Target Protein, was led by Associate Professor Adam Dean, who conducted his PhD within our group and is now the Director of ICU Research at the Royal Melbourne Hospital. This was project managed by myself and Matthew Summers, who is a dietitian within our group, with funding support by the Hospital Research Foundation, and Nutricia provided the enteral formula that were blinded. This was conducted in 6 Australian and New Zealand ICUs and randomised 120 critically ill mechanically ventilated patients to receive either usual protein or a formula that had a higher protein dose called Protein Intense. As part of this, we also conducted a mechanistic sub-study. This was led by Adam Vanson, who was an honours student with our group at the time, and looked at whether microRNA and circulating proteins measured in plasma can predict muscle wasting. To measure muscle wasting, we used a bedside ultrasound to look at the quadricep muscle layer thickness. The microRNA and circulating proteins were analysed at the Imperial College. Adam went over to do these himself in collaboration with Dr. Paul Kemp. And all of these small studies have led us to design our next study, which is going to be a blinded 2-period cluster double crossover registry embedded pragmatic clinical trial, which we'll come up with an acronym for. We plan to do this in 40,000 critically ill patients, so by far the largest critical care nutrition trial in the world. And we're currently seeking funding from the NHMRC with support for formula from Nutricia. This might seem ambitious, but we have had a history of conducting double-blind definitive randomised control trials within our group. The Target trial was published in the New England Journal of Medicine in 2018 and was led from South Australia with Professor Marianne Chapman and Professor Sanji Peek as co-chairs. This was endorsed by the ANZIC's Clinical Trials Group and had funding provided from NHMRC, and Fresenius Kabi provided the formula. This was a 4,000 patient randomized controlled trial that actually recruited much quicker than expected, finishing recruitment earlier than was anticipated. So in summary, just some of our ICU research strengths. I think we're a very multidisciplinary, widely collaborative team. We conduct programs of research ranging from systematic reviews to observational work, complex pathophysiological studies that aren't possible in other units in the world, and definitive randomized controlled trials. We use rigorous methodological design and innovative novel techniques, ultimately leading to clinical translation, which is helped by our clinical academic team. So where will all of this lead? Well, hopefully we don't have ICU patients that leave hospital looking like this, but rather more like this. This is Georgina Harwood, who for her 100th birthday celebrated by going skydiving and later went diving with the sharks, or at least something like this. Thank you.
Thank you, Lee-anne. That was excellent. So again, there are no published questions. So maybe you could just tell us, I mean, that seems extraordinarily ambitious to try and set up a 40,000 patient study. Are those numbers, given that the previous study had 4,000 patients, are those numbers really needed? And how would you go about finding all those patients?
Sure. So the 40,000 patients, to show a difference in terms of mortality, a lot of studies in the past have tried to show ambitious differences in mortality. And we think that in the context of intensive care, where a lot of things can influence that, nutrition may only play a small role, but it's still an important role given most patients are enterally fed when they come into ICU. And at our unit alone, we have 4,000 admissions a year. So when we're doing this across 50 or 60 ICUs in Australia and New Zealand, that 40,000 doesn't seem as unattainable. I guess the benefit is it will also be a registry embedded trial. So a lot of the data collected and a lot of the work that it takes to do a study like that probably is a little bit less than something like done for the target trial where you're randomizing individual patients.
Okay. Still sounds very, very ambitious. All right, well, I think we will conclude our session there. So thank you very much to our speakers, and thank you to the audience for joining us today. So just in conclusion, you can continue to provide feedback on the Q&A area for the next few minutes. That will remain open. Otherwise, you can contact the ABMC on their website. Today's presentations will be uploaded so they can be viewed sometime in the future. And next Tuesday's presentation is focusing on research that's being done in atherosclerosis in Adelaide. Speakers will be Dr. Christina Bursell and Associate Professor Peter Soltis, and it will be chaired by Professor Derek Chu. So thank you again, and we'll see you next week.